All right. Welcome, everyone. I hope that everyone is able to hear. Um, yeah. Welcome to the first session of the 2025 Cancer Genomics Cloud webinar series. I am especially happy to be welcoming Mark to the CGC webinar series because he's been a Cancer Genomics Cloud researcher for several years now, and it has been fun to work with him and watch his bioinformatics skills grow and increase. Mark Primeaux is a PhD student in biochemistry and molecular biology at the University of Nebraska Medical Center under the mentorship of Dr. Punita Dhawan. He earned his BS in biology at UMass Amherst, and his research interests are the biochemical and microenvironmental factors underlying therapy resistance and metastasis in cancer. He was recently awarded the prestigious F99K00 Pre-Doctoral to Postdoctoral Fellow Transition Award from the National Cancer Institute and has accepted a postdoctoral position with Dr. Cyrus Gajar at the Fred Hutchinson Cancer Institute. So welcome, Mark. Please take it away.
All right, thanks, Sarah. Thanks for the introduction. So I'm really excited to kind of talk to you guys about my PhD project. And we'll kind of go into also a little bit about my background and my bioinformatics journey.
Mark, I think you muted yourself accidentally.
Oh, so sorry. Okay.
It's all right.
All right, well, I'll start over. So,
Sure.
thanks. Thank you, everybody. Thanks for the opportunity. I'm really excited to talk about my PhD project, but also talk about kind of my personal journey. We'll go a little bit into my personal background, my non-traditional career path. We'll go into my project a little bit. I'm not going to go into a lot of detail about the science. Just because I know it's a mixed audience, and I don't want to bore everyone with a lot of just Western blots. But if anyone has any questions, please, I can go into as much detail as you guys would like. And if you have any questions, I don't know how you guys usually run it, but put them in the chat. If Sarah is okay with it, you can even ask out loud. That's awesome. And then we'll talk about my bioinformatics journey as a cancer researcher without um, really any background in programming, but just like a huge desire to learn and kind of how I've made my way through that. And then we'll talk about, just touch on, what are my next steps. So one of the kind of most significant parts of my life, especially early life, was moving around. We moved a lot as a family when I was a kid. I've moved around 20 times because I've continued to kind of move around as an adult. And, as an introverted person, this, was really challenging, but also challenged me to adapt to a lot of new places, a lot of new people, and learned how to make new connections. So, there's pluses and minuses there. I lived kind of all over the United States and a few places in Europe. Both my parents are chemical engineers, so they really instilled in me a deep, deep curiosity and really taught me the excitement that comes with learning and that you can find with science and discovery. So it really set the tone for my path. But that being said, my path was non-traditional. I'm a non-traditional student, which is kind of a nice euphemism for saying that I'm the oldest kid in the class, probably by far. Um, I graduated from UMass in Amherst, like Sarah said, in 2009 with a degree in biology. Um, but then, my 20s, like a lot of people, were complicated, and didn't really know what I wanted to do with my life, and got kind of stuck in a rut. And eventually I needed a big major life change to kind of reset things for myself. And so in 2014, I did something totally different. I went and worked on farms. I got an opportunity to move to Italy and work on olive groves and vineyards and just, do manual labor and live a totally different lifestyle. And it was really amazing. It's very humbling because, I couldn't speak the language. So there was just a lot of personal growth there. Um, and through that opportunity, actually got a position as a lab assistant in a hospital where I kind of started dipping my toes back into science, and I helped with data collection and translation from Italian to English of a manuscript that we were preparing, just looking at retrospective rates of HIV and Hep C in drug addicts in Italy. So that really kind of got got me back on the track of getting into science again. And actually I met someone through my experience there that offered me an opportunity to move to Scotland and work as an intern in a lab. So I actually got to get some wet lab experience. That was something I was really lacking. Worked in analytical chemistry, so they gave me an independent project, so I really got to dig into that, and I had some success, and that was super exciting. Um, and that eventually, oh, I gotta mention this is the most Scottish person that I ever met there. His name's Alec, and there's the burn supper that he prepared for me, which is haggis, neeps, and tatties, which is like the traditional Scottish dish. And that's me uh, at the end of my time in Scotland. So I was a real convert. But through that opportunity, I got a job at the Neogen lab in Lincoln. So that brought me back to the States. And there I was working in genomics. And so I was working with gene chips and learning that technologies, sequencing. Um, but after a while, I worked there for, a little over 2 years. And I kind of capped out as a technician. And I really wanted to continue on and get into R&D, start doing some real experiments. But I just didn't have the background or education for it. So so I just kind of like got to a point in my life where, what's the next step? And I was really lucky to have some space and opportunity to go back to school. And I went back to grad school at UNMC in Omaha. I actually started as a master's student because I was planning on just getting a more advanced degree and then going back to industry. But I just got so into my project and just kind of found my place in that world and really fell in love with research and switched to a PhD within about 6 months. I mean, it was pretty quick. So we'll kind of go over what my project is. I'm studying the role of Claudin-1 in colon cancer. Probably a lot of you don't know what Claudins are. I certainly didn't when I started. But they're a really important component of the barrier in epithelial tissues between the outside world and your body. So they're kind of at the top of the different junctional complexes between epithelial cells, and they're what actually stitch cells together to really control what flows in between cells to make sure that things aren't leaking out of your body, right? Um, so this is the structure of the actual protein itself. So it's like most proteins, it's one long chain basically, but it passes in and out of the membrane 4 times. There's an extracellular loop, or there's two extracellular loops, sorry. And these kind of help mediate the interactions between claudins onto neighboring cells. So that's kind of what forms that tight bond. But there's also this intracellular tail um, that hasn't been as studied, but it ends in this really interesting amino acid motif at the very tip called the PDZ binding motif. And I'll be referring to that throughout the talk because that's really important for interaction with other proteins. So in normal physiological conditions, it helps to kind of build a network of proteins that support and strengthen this junction between cells. And what I'll show is that in cancer, you start seeing weird interactions with other proteins that have different effects. So they're more and more they're being researched as potential therapeutic targets. This is a list of just recent clinical trials. So there's a lot of different strategies. There are monoclonal antibodies, other immunotherapy approaches like CAR T cells, and then people are designing small molecules to bind to claudins to kind of affect their function. But what makes them good targets? So there's a lot of them. There's 26 different family members in humans. And what's important is that a lot of them have very tissue-specific expression. So what this means is that, one claudin may be expressed primarily in the liver, while another claudin may be primarily in the skin. So what this means is that you can target specific ones and kind of limit off-target effects. You're not hitting all the organs in your body when you're targeting a particular claudin. They're also at the surface of the cell, so they're available for drugs to bind, like antibodies or inhibitors. And certain ones actually show increased expression in cancer. So this is just one example. So this is colon cancer. This is what we study. Now, in normal, healthy colon tissue, you have high levels of claudin 3. So that's what forms that tight barrier I was talking about earlier. But you normally have really low levels of Claudin-1. What happens is during cancer progression, you actually see a flip. Um, so Claudin-3 starts to go down and Claudin-1 starts to increase. At the same time, you see a breakdown of just overall organization of the cell. So normally, they're all up in this apical junction, but as the cell starts losing its kind of colon cell identity, becomes less differentiated and less organized, you start seeing expression all over the surface of the cell. So this is just another way of showing that. So in a healthy cell, everything's kind of gathered right here. These are your claudins, and these are your other proteins. Everything's nicely segregated. But in tumor tissue, again, everything kind of gets mixed up. It's a little bit like chaos. And what happens is that as these abnormal interactions start to happen, you start seeing changes in signaling in the cell. And we see evidence of a lot of changes in important survival and oncogenic signaling like AKT or Wnt beta-catenin. Um, but the question is, what are these intermediate mediators? Because claudins by themselves are not chemically active. They're not enzymes, so they're not catalyzing chemical reactions by themselves. So they have to be acting through another enzyme, another receptor, for example, to make cause these changes. Another cool thing is that they're really good targets for drugs or antibodies because in healthy tissue, the antibody simply can't reach, it can't find the claudin. It's kind of sequestered away in these junctions. So that means that it doesn't there's limited binding to healthy cells, but it tends to preferentially bind to cancer cells because they have these exposed areas and they're able to find their target. And so the idea is that, either with an antibody, you're triggering an immune response, or you're actually affecting those protein interactions and, causing effects in signaling. And another project that we're working on is taking those similar antibodies, tagging them with a fluorescent tag so that you can put this in a patient. You know, we've done this in mice, but you can put it in a patient and it'll bind specifically to the tumors. And so a surgeon can then go in there. The tumor is actually glowing, and they can find the margins of the tumor to be able to resect it more effectively without leaving anything behind. So why are we even studying this? So the problem is that cancer has both intrinsic and acquired resistance to the therapies that we have. So basically, with colon cancer, for example, if you catch it early, you have a pretty good chance of survival, over 90% at 5 years. But if it's already spread to distant sites, that plummets to around 13%. And that's because we basically don't have systemic therapies. I mean, cancer is really good at adapting to insults. And so almost always the cancer develops resistance to whatever chemotherapy we're using. And the conceptual idea is that with chemotherapy, you're killing, you kill 99% of the tumor, but there are some intrinsically resistant cells within that tumor that have more stem-like qualities that can survive and then eventually regrow a more resistant tumor. Now, what our lab has shown previously is that not only is Claudin-1 expression kind of a sign of cancer progression, it actually itself promotes cancer progression. So if you take a cancer cell and overexpress Claudin-1, you actually make that cell more aggressive and more metastatic. And the opposite is true if you knock it out. So it's actually changing the behavior of the cell. So that was the rationale for my study. So we see that Claudin-1 expression is associated with all these characteristics of aggressive cancer. Um, these specific characteristics are associated with stemness and resistance to therapy. And so my question was, the next logical step is Claudin-1 promote chemoresistance? And like the deeper question that I was more interested in actually was how was it doing it? You know, what was the actual mechanism there? So this is kind of a summary of my findings. Um, Kind of give you a roadmap. So basically, we have shown that if you throw Claudin-1 into a cancer cell, it stabilizes this receptor kinase called FA2. And FA2 is a signaling molecule. And when it's stabilized, it activates survival signaling in the cell, and then that is what is increasing chemoresistance. So that'll kind of help you understand as I kind of run through the data. So first, we looked at this really amazing data set from the Broad Institute in Boston. So they took 62 different cancer patients who had colon tumors, and they took also kind of adjacent normal tissue and used single cell sequencing to look at the gene expression profile of every single individual cell in those tumors and their normal tissue. And what we can see in that data is that high Claudin-1 expression is overwhelmingly associated with just the tumor cells. You barely see any expression in the normal healthy tissue. So seeing that level of differential expression, one says that this could be an important therapeutic target, and two, this could be associated with the cancer progression itself. And this is a whole range of patients representing different genetic backgrounds and different cancer subtypes. When we look at the individual, no, this is not spatial single cell. This is just normal single cell. And when we look at the characteristics of the individual tumor cells, we see that Claudin-1 expression is associated with the more stem-like tumor cells, the more proliferative tumor cells. So this really supported our hypothesis. And when you look at what are the gene expression patterns that are correlated with Claudin-1, we see upregulated pathways that are really important for chemoresistance, like mTOR signaling, E2F targets, MYC, P53, apoptosis. So this is really good preliminary data. We looked at another patient data set where patients were given chemotherapy and then well, they had their tumors sequenced, and then they were given chemotherapy, and then they were divided into people who responded and people who did not respond to the therapy. And we found that tumors that had high Claudin-1 expression um, were particularly resistant to FOLFOX therapy, which is kind of it's a combination chemotherapy. It's a standard of care for colon cancer, and it's three drugs: folinic acid, 5-FU that I'll be talking about a lot, and then oxaliplatin. So now that we had that preliminary data, we wanted to actually look at biochemistry. Like, is there a causal relationship here? So we took colon cancer cells, and in one cell type, we overexpress Claudin-1 artificially. And here we saw a fourfold increase in its resistance to 5-FU, that chemotherapy drug. And in two other cell types that already express Claudin-1, we knocked it out with CRISPR. And here again, we see the opposite. We see a 2.5 and 6-fold decrease. So resistance to 5-FU is related to the level of Claudin-1 expression in these colon cancer cell lines. We also did this in mice. So we gave mice tumors that either expressed Claudin-1 or tumors that had that we used CRISPR to knock Claudin-1 out. And what we found was pretty significant that even with 5-FU, if there was Claudin-1 in the tumor, it just continued to grow bigger and bigger, even with the treatment, almost to the same degree as the untreated tumor. Whereas if we knocked out just Claudin-1 with CRISPR, it became much more sensitive. And as a biochemist, I wanted to understand what specifically was going on, what was mediating this on a, molecular level. So we took two Claudin-1 constructs. One is like the normal wild type full-length Claudin-1 and compared it with a mutated version that had that PDZ binding motif deleted. And our hypothesis was that whatever protein was interacting with Claudin-1 might be interacting through this amino acid sequence. And what we found was that the full length, but not the mutated version, was what conferred resistance to 5-FU. So the next question, what's the actual signaling pathway that is being affected? There are lots of different oncogenic signaling pathways that are going on in the cell, but what is specifically being regulated by Claudin-1 in this system? So we took an overexpressing cell line and then a knockout expressing cell line. And we used an array to look at changes in 37 different pathways. And we found that the AKT mTOR pathway was reciprocally regulated in both of these systems. So when we overexpress Claudin-1, we activate AKT. And when we knock it out, we see a decrease. And AKT is a really important molecule for cell survival. and for chemo resistance. It basically just sits there and tells the cell to just keep surviving no matter what. And importantly, we found that when we mutated and cut out that PDZ binding motif, we lost the activation of AKT there. So again, this is saying that it's being mediated through this mysterious protein interaction. So just to kind of summarize what I've shown so far, Claudin-1 increases stemness and chemoresistance via that PDZ binding motif. It activates AKT, the survival signaling in the cell. But again, we come to the question of what's in between. Like, what is that intermediate effector there that we're missing? So we did a screen. We took three colon cancer cell lines and looked at all the proteins that were interacting with Claudin-1 in all three of these and compared them. So we had one cell line that is more like your normal cells. Everything's kind of intact. You have your normal, everything's organized. So you would expect to have all your normal interactions there. And we had two other cell lines that represent more advanced cancer. Here, all the junctions are kind of dissolved, everything's mixed up. Here we would see those more abnormal interactions. And so we focused on this, let's see, on this this subset here. So these 70 proteins that interacted in these two cell lines, but not this one. And from that, we took four different pathways or four different molecules that were known to affect AKT and used drugs to inhibit each one. And out of that screen, we found that when we inhibited FA2, that was the one that kind of returned AKT activation back down to a similar level as if Claudin-1 wasn't there. And importantly, FA2 interacted with Claudin-1 in those two cell lines that had disorganized junctions, but not in the CACO2, the more organized cell line. So again, FA2 is actually a receptor kinase. So this is a molecule that is chemically active that specifically can affect the chemical reactions that drive this survival signaling. So we validated this screen by using immunoprecipitation in all of our cell lines. So here you can see if there's a band here, that means that if you grab Claudin-1 in a mix of the cells and pull it out, you're also going to pull out FA2 along with it. That's basically what this is saying. And importantly, we found that in the mutated version, we don't see the interaction. So you see it when you have the PDZ domain, but you lose it when you cut it out. So we're not seeing the interaction with the mutant. Next, we looked at the connection between Claudin-1 expression and EphA2 expression, first in our cell lines. So in our overexpression cell line and our two knockout cell lines, we saw that anywhere where you have higher Claudin-1, you have higher EphA2. So you can see it pretty clearly here. And anywhere when you knock out Claudin-1, like, sorry, right here, you see EphA2 going down. So there does seem to be a relationship there. We have a collaborator, Dr. Fisher, here at UNMC, and he's a pathologist and he gets samples from different patients, and we can cultivate these tumors as organoids. And we see that in organoids that have high Claudin-1, we tend to see significantly higher EphA2 in these compared to our Claudin-1 low organoids. So what's going on here? How are the expressions connected? So, my sort of working hypothesis was that these are actually physically interacting. And so maybe Claudin-1 is almost holding EphA2 hostage, right? Because all proteins in the cell kind of go through this process of being produced and being degraded. So there's this constant cycling and recycling of products in the cell. And we can actually capture this by using a drug that stops protein synthesis, this cyclohexamide. So we can stop protein synthesis, and then we can take different time points after that and see how long does any particular protein last over time? How stable is it? And, without Claudin-1, we see that EphA2 is pretty much gone by 24 hours. But if we add Claudin-1 into those cells, it's much more stable. You can see after 32 hours, it's basically the same as it was at 0 hours. So basically, when Claudin-1 is there, it's holding on to EphA2 or Fa2 and keeping it from being physically broken down. And so it's basically sticking around in the cell longer and is able to signal for longer periods. And as you can see, we don't see that stabilization with the mutant here. And finally, to test that it's actually being like what the phenotype we're seeing is being mediated through FA2, we then took our Claudin-1 expressing cells and then knocked out EphA2 with CRISPR. And here we saw that one, we lose AKT activation. Two, we see a drop in resistance to chemotherapy. And three, when we went back and looked at that patient data set, we found that in patients that had high expression of FA2, they were particularly resistant to Folfox. So a similar pattern to Claudin-1. Now, I'm a visual person. I like to understand how things fit together, And so we use another really amazing tool that's been developed over the last few years called AlphaFold, um, which uses machine learning and artificial intelligence to predict how proteins actually fold in three-dimensional space and how two different proteins can physically or can interact. So we modeled the C-terminal tail of Claudin-1 along with the intracellular domain of FA2, and we found that it predicted it binding right in the active catalytic pocket of FA2. And when we zoomed in and looked at it at an atomic level, we found several amino acids that were stabilizing this interaction. So the next step, yeah, this is great, but it's just a prediction. This is a mathematical prediction. So we wanted to test it and validate it. And what we did is I took these, I found some certain amino acids that I predicted were actually stabilizing the binding based on their, chemical characteristics. And I mutated those and checked the binding in my cells and found that when we mutated those amino acids in the catalytic pocket, we lost the interaction. So again, this kind of validates the power of these machine learning algorithms to predict real interactions. So that's kind of the conclusion of a very quick and kind of 1,000-foot view of my project. So basically, the parts that we've added here to the conclusions are that Claudin-1 binds FA2 through those intracellular domains, and it increases its expression by stabilizing it. And that, in turn, is what's actually activating the survival signaling and leading to chemoresistance. So, we've got a lot of interesting future directions that we're taking. Like, how do we target this? Can we use inhibitors and antibodies to target this and actually counteract that chemoresistance? But now I kind of want to talk about my bioinformatics journey. Because I'm a cancer researcher, I didn't have any training in bioinformatics. I went to school, went to undergrad in 2009, probably before bioinformatics was even a word. But and I never really had any programming experience. So I really struggled to understand a lot of the research that I was reading. I was kind of desperate to get a toehold and because I knew that I needed these skills to be able to have a career in this field. So in 2023, I kind of responded to, it's one of those millions of emails that you get from your department saying, hey, there's this workshop if you're interested. And I looked into it, and it's called the Big Data Training for Cancer Research. And it was exactly what I needed. It was tailored to cancer researchers who had minimal programming experience, but who are ready for a very, intense, hands-on 10-day, all day uh, workshop that went into both the theory and practical skills. Um, and I mean, it was absolutely incredible. I would highly recommend it for cancer researchers of all levels. I mean, there's all levels in there, grad students, postdocs, professors, all levels. Um, and you really leave there with some usable skills. And best of all, it was free, which not very many things are that are worthwhile these days, but it's all NCI funded. So as long as you can get there, and now it's at UC Irvine. When I went, it was at Purdue. But there are travel awards as well, but I paid for my plane ticket, but once I got there, room and board, food, everything else was covered. So I felt really, really lucky to be involved with that. But through that workshop is how I found the CGC. And I was really excited about it because one of the big barriers of doing bioinformatics analysis is that these files are huge. You need a ton of computational power to be able to process all this data. You either need a dedicated high performance computing system, access to a supercomputer, for example, or these days more and more that you can access cloud computing, um, which was like shockingly accessible, even for someone like me that didn't really have any experience with computing beyond, your standard desktops. But so through them, I learned the basics of cloud computing, data management, analysis pipelines, and I'll kind of go through that. But yeah, so what I want to say first is that one of the reasons I reached out to the CGC is because I had this problem that there was all this publicly available data that's freely available, but there wasn't an easy way for me to access it, and there definitely wasn't an easy and straightforward way for me to process it into any form that was really usable for me and interpretable for me. And what I mean is that the NCBI, which is a part of the NIH, has this incredible resource called the Sequence Read Archive, which allows people to upload any of their sequencing data to the NIH to make it publicly available for other researchers. And a lot of grants these days require that any research that you do with sequencing that you publish, you're required to make it publicly available for other people to use. So there's all this just an unbelievable amount of raw data in this archive. So you have all these collections of different studies. Um, Available. And one of the cool things is that you can actually find, you know, a bunch of similar studies. They may have been doing something, a slightly different problem, but it's a specific tumor type or a specific treatment. And they did all the sequencing and put the data on there. And you could find all these different studies and actually combine the data and combine all of those different patients or mice or whatever to make your study that much more robust. But the problem is that all this data is in its rawest form for the most part. And so there's all kinds of steps that you need to take to be able to use it. You know, you have to download it. These are big files. They take a lot of time and a lot of space. They have to be checked and pre-processed. Um, all the reads have to kind of be mapped back out to the genome so that you can say, oh, this particular gene has this many transcripts, and then you can kind of correlate that to how much is this gene expressed overall. And then it has to be kind of processed through a statistical model to be able to make any sort of real comparisons uh, for to kind of address your experimental question. So there's all these different steps along the chain. And the problem is that, each one of these components has, a bunch of different programs that have been developed to tackle each of these different problems or each of these different steps. And they were all developed independently. And so it's kind of hard to tie them all together, right? Because what comes out of this is a certain file format that may not be compatible immediately with this. So you have to convert it and you have to put it into different formats. And so everyone that does this professionally kind of has developed their own pipeline that they use, which requires a lot, you know, it's not the simplest programming. Like you have to be able to manipulate large data sets. You have to be able to program um, at a higher level to be able to do that. So there wasn't a standard plug and play pipeline. So that's that's kind of what I approached the CGC about. I was interested in designing a pipeline that would be more of a plug and play, at least for my analysis, and maybe even make it available to other people for other people to use. And they were awesome and gave me funding from the NCI to kind of do this processing and do this whole learning process. And I received funding, but I also got a ton of technical support because I'm still new with all this. I'm still new with even just like the conceptual part of it. Um, and I will say there's nothing like revolutionary about the pipeline I developed, and someone who knew what they were doing could probably do it, relatively quickly. But for me, it's about the learning process, and people like Sarah and Rowan were amazing and supportive in that. But the CGC had these individual components, and we figured out ways to tie them together and to move the data in a way that was more seamless through the whole pipeline. See. And basically, um, briefly, you just put in a list of your sample names. They don't have to be from the same study, just a list of sample names from all different studies at the input. You put in a few other files that are standard on the CGC. You tell the program what you were comparing, in this case, like comparing different tissues. And then you get output that you can use. So you get all your gene counts, you get all your comparisons. It's pretty, it's really straightforward. And I was kind of surprised this didn't already exist. Um, and one of the huge benefits of cloud processing is that you can do things in parallel. Because if you had to process all these samples one by one with one computer, it would take days. But with cloud computing, you could take each of the samples and split them off into individual computers that process everything in parallel. And so you can kind of see this represented here visually. So they're processing all these at the same time instead of one by one. And so kind of learning how to do that was really cool, learning how to set it up to do things in parallel and just seeing how much more efficient it got. And I'm sure like, the experts could pare that down even more. This is just a snapshot of one of my initial analyses. I took two data sets. One had three patients, one had 18, shoved them together. And like here, we can look at Claudin-1 expression comparing primary tumor to normal tissue or metastatic to normal tissue, right? All these dots represent different genes. And so all of these are potential new projects. You can see like, oh, this gene goes up in primary tumor and even higher in metastatic tumor. This may be something to look into. And so, this has turned into a data set that, my lab will continue to use in the future. Um, right. So what's next for me? Like Sarah mentioned, um, I was extremely lucky to be awarded the F99K00, which is a transition grant that supports the end of your PhD and then also 4 years of postdoc. And so the idea is that if a postdoc mentor doesn't have to pay for you, then it's going to be a lot easier to find a good position in a good lab. And I was lucky to get an awesome postdoc in a lab at Fred Hutch in Seattle. And the idea is that with my doctoral project, I was looking at things that are happening inside the tumor cell that cause it to be more aggressive. In my postdoc, we're going to be using all kinds of different modalities to figure out how the environment that the tumor cell is in affects its behavior. So what we're looking at is why do tumor cells grow in one metastatic site, like the lung, but not in another, like skeletal muscle or the spleen? What is it about those environments that affect whether you're going to get a tumor or not? And then can you use that information to then try to prevent metastasis in general? So I'm super excited about that. Um, I feel like the foundation that I've made for bioinformatics so far is absolutely crucial because now I'm going into spatial sequencing, spatial profiling, um, AI tissue classification. It's going to be a lot more intense and it's just going to build on, the knowledge that I have gotten so far. So I don't know. Overall, I just wanted to kind of present this as kind of a case study for someone who maybe, 6 years ago, 7 years ago, definitely wasn't on the path that I'm on now. And I didn't know anything about bioinformatics, didn't know that word existed. And now, I've there are steps that you can take to kind of build that strong foundation, and it really, really, really helps uh, in your career. Like it's so necessary these days. But yeah, that's my talk, and I just want to thank my lab and my mentor, Dr. Dewan, and everyone who's helped me along the way. Especially a shout out to Sarah and Rowan here, who've been absolutely awesome with this project, and just like they've taught me so much in general about, you know, bioinformatics and sequencing and analysis in general. But let's see, that's all I have. So I'll take any questions anyone has.
I'm going to stop our recording so that people who did not sign a release form are free to ask questions. But thank you so much, Mark. This was wonderful. So recording